This chapter is about phylogenies or phylogenetic trees. Those are basically pictorial representations of evolutionary relationships between organisms or organismal groups. So here's an organism we're looking at, and if you had to have somebody guess what this was, you'd probably get the answer of a snake. It has a lot of characteristics that look like a snake, including no limbs, a long cylindrical body, it has scales, no external ears, it does have a small eye. Here's another species of the same sort of organism. You'd probably say that's a snake. These two as well. In fact, you can even see a forked tongue, which we think of as a snake characteristic. But in fact, all of these are actually legless or limbless lizards. Some of them actually have these remnants of limb buds on their bodies. Some of them have no limbs at all. But here's a true snake, and there are actual differences phenotypically between those that you might not see at first glance. So you can see the head looks kind of different, right? The vertebrae are different in the necks of snakes and these legless lizards. Legless lizards have eyelids, whereas snakes don't. And sometimes, like I mentioned, they have limb buds where none of the snakes are going to have those limb buds. And so what this is showing us is that leglessness is not a feature that evolved once. It must have evolved more than once. And here, even within these lizard groups, I am showing four-limb phylogeny. So this is what a phylogeny is, or a phylogenetic tree. It's this kind of branching sort of structure like a family tree that shows relationships. So this is the phylogeny showing the four limbs, and here's the key showing you how many digits it has, or limbless. So we're looking at the blue ones here. And this is the hind limb. So this is the same phylogeny, just highlighting which ones have different digits or limbs. So we look at the ones that have blue on both sides. Those are completely limbless. Whereas something like this guy here with the blue on one and the orange on the other would have hind limbs with 2 digits and 4 limbs without, or no 4 limbs at all. And so what I want you to notice is that limblessness does cluster. So all of these groups, all of these species are limbless. But it's all over the place in this lizard phylogeny, multiple places. It's not all the blues grouped together. And so what that means is that leglessness evolved multiple times. And usually this is an adaptation to similar living conditions or an adaptation that allows a particular behavior that's advantageous in certain environments. When we talk about phylogenetic trees, you probably heard classically of this kingdom, phylum, class, order, family, genus, species classification system that was developed by Carl Linnaeus in the 18th century. And he had this idea of a hierarchy of organisms. Now, the branching pattern is actually more accurate because it takes into account ancestry and things like this we understand better now. He more thought it was sort of like a ladder with certain organisms at the top and other ones at the bottom. But his naming conventions are still used today in a large part. And so you can see that we're going to start large and get more specific as we go up this diagram here. So we have 3 domains that we use currently, the archaea, eukarya, and bacteria. And you can see here that this leopard is in the domain eukarya. There's multiple groups within that, and it's in the kingdom Animalia. Within Animalia, there's multiple groups within that, and it's specifically in the phylum Chordata within Animalia, and then so forth. So you do need to know domain is the biggest, and then you want to know kingdom, phylum, class, order, family, genus, species. You need to know the order of that, and this is from broadest to most specific. In the end, a species is only one particular type of organism. Just to point out, mostly we'll be looking at genera. That's the plural of genus. So we'll be looking at genera. We'll be looking at some species, and a lot of phyla, or phylum is singular, phyla is the plural version. So that's what we're looking at. And any of these groupings can be called a taxon, or taxa is plural. And so this just means a grouping in a hierarchical order. I want you to notice there's specific ways to write these words, and you do have to do that correctly on lab practicals and things in class. So we're looking here at a species name. So species names are always binomial. Bi, of course, meaning two, nomial meaning names. So they have two names to them. So sometimes people think this last part here is the species, but in fact, it's actually the whole thing. So Panthera pardus is the species name. And you'll notice here it's in italics. Okay, so you have to italicize both parts of the species. If I was writing it, I'd underline that if I was handwriting it. The first part of the species is actually the genus. And if we look back here, you can see Panthera is the genus. So the genus is capitalized, and that is underlined or italicized. And the epithet, specific epithet, which is the second part, is also underlined or italicized, but it is lowercase. So if we were going to write, and you could take a second to do this, the correct way of saying Homo sapiens, which is what modern humans like us are, you could pause that, but we would write it like this: capital H, O, M, O, underline, and then separately, lowercase s, sapiens, and we'd underline that or italicize if we were typing. You can also just write the genus. If you're writing the genus like Homo, that's the genus of hominins, so that's like Homo neanderthalis, the Neanderthals, and Homo habilis, Homo erectus, Homo sapiens. So all of those are in the same genus, and you would make that capitalized and underline it. If you were to use any of these other groups, those are not italicized. They would be capitalized though. So for example, if we were saying Homo sapiens are in Animalia kingdom, that would be Animalia. But if we're just calling them animals, of course, that's lowercase. That would be. But if we're just calling them animals, like the generic name, that of course is not capitalized. But if we're calling them just generically animals, not Animalia kingdom, that is not capitalized. So something like Chordata, which is a phylum, is capitalized, and chordates, the generic name, chordates is not. But none of those are underlined. So you do have to write it correctly. So here's a phylogeny with your groups superimposed on it, and sometimes it lines up to where the order, family, genus, species levels are also mostly lined up with the branching. Sometimes it's not. And so what that indicates to us is, are these groupings actually valid if the Panthera existed here and then another, quote, Panthera is over here, if they're not actually in the same evolutionary group, is it valid to even call them both Panthera? So that's something that's being argued right now. What these trees show are hypotheses. So you'll actually notice if you look at different editions of the textbook recently, or papers that come out, there are new phylogenies being published all the time. And that's because we have additional data. Phylogenies are built based off of data, so different groups could use different data sets to build the same type of phylogeny. They could use different parameters, so you have to decide parameters, and if you change those, sometimes that groups them a little bit differently. And so the different lab groups will put out their phylogenies, explain why they think this is the best way to do it, and other groups will either agree with them or argue against it, and eventually that scientific process leads to phylogenies that are more or less accurate. So I will indicate to you sometimes when we're not sure, we're like very not sure about relationships, and we'll indicate that on our trees. And there's other trees that are very well established that we feel very confident in the relationships there. But they are hypotheses, and we are willing to change trees if new information comes along. So here's an example of a tree. You need to know these parts. A lot of this is just sort of definitions. So The tree is being read in this case. We're looking at it and reading it from left to right. This is generally how I draw my trees in class, but they can be drawn a number of ways. This tree is what we call a rooted tree. That's because we are drawing the common ancestor of all the groups. So this ancestral lineage, this ancestral organism type here's the root. That's the root of the tree right there. The other organisms are the ones that we're looking at here. So taxon, remember, that's just a group of organisms. And so here are our different groups. The one that branches first is called the basal taxon. Basal means bottom. So it doesn't mean it's worse or better or less or more evolved. It just means that it branches off. So here's a branch point. You'll notice that if we're walking along in time this way, this is the first one to branch off. And the taxon G. So that's the basal. Sister taxa are ones that share a common ancestor. So ancestor number 4, that makes B and C sister taxa. There are other features of the tree that you need to know. I mentioned branch points already. Branch points represent last common ancestors. So the last common ancestor here, there, here, and here. And there are organisms, ancestors all along this line. What I like to think of is that there's one type of organism, one group, and then some of that group, generation after generation after generation, turn and change in a certain way here that eventually leads to taxon G today. And the other part of the group changes in different ways until it leads to this common ancestor type 2, which then it diverges. And generation after generation change in one way, generation after generation change in another way. So all along these branches are different generations of organisms, and they could actually be different ancestors, different species as we go along. But we don't always draw them in. There are trees that do. Sometimes we are just mostly concentrating on modern day organisms. And you'll notice that this looks different here, right? It's called a polytomy. Poly means many. So this is when you have multiple groups all branching from the same common ancestor. So it's highly unlikely that 3 groups diverged all at the same time. Probably what happens here is that one group's going to diverge and then the other groups would split later. But because we don't have enough data to help us resolve that, we just leave it with multiple branches. You can even have polytomies that are bigger than that. Here's 4 polytomy, and we'll draw these in class. So there's a really good website, Understanding Evolution. I have this linked to on your module. I'm just going to show the little video here. So this is showing you a bunch of different types of trees. These are all actually representing exactly the same thing, but you can draw them in a number of different ways. In the end, all you're looking at is the branching pattern. Some trees are more or less informative or more or less confusing. What I'm going to mostly draw is this type of squarish tree. So this type of tree here, the squarish. You probably also need to be able to read this kind of diagonal tree. And when you go here, when you go online and look at this, you can select all the different types of trees, and you can click on each of these and they'll tell you how to read that type of tree. You can also click on the advanced features, and that's going to actually give you more information about different versions of that tree and how those would be made. If we look at a tree, let's see if we can figure out what's going on. All right. So in this tree, my question is, which one have evolved the longest? We're looking here particularly at shark group and mammal group over there. You can pause the video. Okay, so if we're looking here, most people are probably going to say sharks if they don't think about it too much. So, you look at this and you're like, man, sharks have this really long branch. They branched off earlier. That must mean that they've been around or evolved longer. And also shark fossils, they tell you, show up back here 440 million years ago. Whereas mammal fossils don't arise until 160 million years ago. But if we actually look and we trace the tree, that's what you're going to do. You're going to walk the tree to sharks. So you'll notice the distance you went was from here to there. And you'll walk the tree There's more branching for mammals, and the distance is here to here, right? And they share the last common ancestor back here. And so really, mammals and sharks have been evolving for the same amount of time. Mammals have not been around for that long period, for all that period of time. These were mammal ancestors that were not mammals or reptiles. These were ancestors, mammal ancestors that were some sort of early four-legged land vertebrate. These were ancestors that were before the split of fish and mammals, but all of these are mammalian ancestors, so they're evolving that entire time. They're just not in mammal form. In fact, all modern species, extant means ones that are living today, they share the same common ancestor and then have arisen from that. Every organism on Earth today all have 3.5 billion years of evolutionary time. So then we can look at these and think which is the most evolved group. A lot of times I would draw phylogenies like this to make them a little bit easier to remember, where you just have each group branching off to the left or to the right. So sometimes you'll look here and you're like, wow, look at this stair step. Here's mammals at the end. They must be the best, the most evolved. And look, they branched most recently, so they must be more evolved than, let's say, lampreys, which branched a lot earlier off. But really, all these trees are equivalent. You're just following the branching pattern. And if you look at that, lampreys branch off first. Lampreys branch off first. Lampreys branch off first. And then we go to sharks and rays branching off, and then everybody else. Here's sharks and rays branching off and everybody else, and here are sharks and rays branching off and everybody else. And if you just keep following that, in the end, the last branch is the lizards and the mammals. Lizards and mammals, lizards and mammals. And because the branching pattern is the same, all the groups, all of these are equivalent phylogenies, and none of the groups are more evolved than others. So sometimes it looks like this is a goal of making a mammal, but really it's not. It's just that we drew the tree that way, that it looks like, a stairway. So there's really not a main line of evolution. There's no goal or pinnacle, and no organism is more or less evolved. They're just differently evolved. This is also a tricky thing, is sometimes it just depends on what you've chosen to put on your phylogeny. So here, you'll notice that flowering plants branches off first, and then we have the rest of these, and we've expanded this group. They're called gymnosperms, and that's another type of plant. So you might think, wow, flowering plants branched off first. They have a really long linear, or a long line that doesn't change at all. Now, they must not have changed in time. But really, that's just because we chose not to branch them out. In fact, here's flowering plants. And now in this one, same phylogeny, but we've just expanded the flowering plant group. And if you looked at this, you probably wouldn't have that same misconception that flowering plants have undergone less change than the other groups. So you just have to be careful not to overinterpret the phylogenies. It also doesn't matter which one is on the left or the right, or the top or the bottom. That has nothing to do with each other. All that you're looking at is the branching pattern. So the ones that share the most recent common ancestor, they are more related. So like here, pines and neophytes are most closely related in this phylogeny. And then if we look at cypresses, who's their most close common ancestor, so you look back in time, here's the last common ancestor we've drawn here, and then you say, well, who is branching off of that? Pines and neophytes. So cypresses are equally closely related to pines and neophytes because they share the same common ancestor. Who is most closely related to flowering plants? Well, if you think about that, go back to their last common ancestor, and then it turns out that everybody in this whole phylogeny is equally closely related to flowering plants. This is an important point here. It should not be assumed that taxon evolved from the taxon next to it. For example, so B and C are modern day organisms. B did not evolve into C. A did not evolve into B or C. They share a common ancestor. That was an organism that's different than A, B, or C. So a lot of people have a misconception here with the ape phylogeny. So I'm going to leave out some of the groups. All right, and there's actually a split with chimps and bonobos too, but I'll just draw it like this. So sometimes people make the claim, right, like, oh, humans couldn't have evolved from chimps. That's too weird. They're right that humans didn't evolve from chimps. Humans and chimps shared a common ancestor back here that was neither human nor a chimp. It was something different. And then some of that group started changing in one way and became modern day chimps. Another part of that group started changing in a different way and became modern day humans. So it's not like they turn into each other. So to build these trees, we use a number of different things. One that they used to only have is morphology, so that's how they look, which works sometimes, but there are other times when morphology is going to lead you astray. For example, if you made a tree and put all the legless reptiles in it, that would actually be an incorrect tree because you'd have snakes and lizards, legless lizards, more closely related than legged and legless lizards, which should be grouped closer together. So we also use data that's looked at through molecular testing, so things like gene sequences, sometimes protein sequences, and the more closely related the organisms are, the closer we expect their sequence to be. So if you had an earlier branching organism or group, you'd expect that the DNA sequence would have accumulated more mutations than B and C, which have only had this much time to accumulate different mutations. So here's the mutation accumulation time. And so looking at trees helps you understand if a feature is homologous or analogous. So homology, homo means same. Remember, so homologous means it's a feature that is shared due to common ancestry. And analogy is a feature that you see due to frequently like common environment or some sort of conditions. The leglessness that we saw would be analogous. Here's an example of homology. So we have a branching pattern here, and tetrapods means 4 limbs, tetrapod. So you can see the tetrapods all grouped together. So all of these are 4 limbs. The ray-finned fish do not have limbs, or the sharks. And so we can put that feature on the tree here, and it's before the branching of all the tetrapods, which means that that is a homologous feature of all tetrapods. So the origins of having 4 limbs comes from the common ancestor. Other examples here. So here are bats, mice, birds, and crocodiles. So all of these have 4 limbs. So 4 limbs is a homologous feature of bats and birds, let's say. Whereas having wings, bats have wings, but they're more closely related to mice. Birds have wings, they're more closely related to crocodiles. So that means that having those 4 limbs modified into wings is an analogous feature. I mentioned that besides using morphological features, sometimes we use molecular sequences and such to figure out if organisms are closely related or not. This is one of the most robust ways to do it. And so here are 2 nucleotide sequences, so this would be DNA. And you can see there are certain areas that are similar, and then there's ones that are different. So the similarities would be marked with a star, and the differences would be left blank. Okay, so then the question is, of course, this is a very small sequence, a short sequence. This is from organism number 1, and this is from organism number 2, and we say are 1 and 2 closely related to each other. And so it's, I don't know, it's kind of hard to tell with your eyes. And so we usually use computers to look at sequences. It's much longer sequences as well as multiple sequences to make it more accurate. There are parameters that have to be run in order to have the computer compare the sequences and align them. And what the computer is looking for is what we call parsimony, or maximum parsimony, which means that there are the fewest number of changes based off your branching pattern. And they have examples of this in the book. It's sort of Occam's razor. The most likely scenario is that the least amount of changes occurred. So it's less likely that a sequence arose multiple times all over the tree. It's more likely that that sequence was here and then all the descendants share that sequence. So we mentioned trees are hypotheses, and you use a combination of data to see like what actually makes sense. And there are two types of changes that are most frequently looked at for trees. They're called SNPs and indels. So when you're comparing sequences, first of all, you have to compare the same genes. We call those homologous sequences. So obviously they're not going to be similar if they're completely different genes. So you find a gene, and then you're going to have the computer do it, the comparison. And here's a little example of alignment. So here's some sequences, and the computer will take them along, and it would be longer than this, and match up. And each time there's a match, it detects that. And so basically it tries putting blanks in a bunch of different permutations, and it sees which one gives it the most asterisks, let's say, which means the most similarity. So the two types of alignments that you see here. Here's your SNP. So a SNP is actually a single nucleotide polymorphism, which just means single nucleotide, one nucleotide. Polymorphism means that there are many morphs or types in the population. So it's just one base change here, basically a mutation that occurred. So this sequence on the top, these are not paired together. The sequence on the top is one organism. The sequence on the bottom is another one. So that would be a SNP. The other type of change that it detects is called indels, or insertion deletion. So here's an indel. So it's either that the A was inserted in that organism on the bottom, or that the A was deleted in the organism that you're comparing on the top. And so that gap there actually, you see, makes the sequence align better by adding that gap in, because then the G and the A line up, whereas if you didn't add that gap, you wouldn't have those aligned. And so the computers can detect that. So here's 3 different sequences being aligned, and you can see obviously the more sequences you have, the more complicated it will be to figure out the best alignment. And so in the end, the ones that are most similar to each other with the least number of changes, the least amount of SNPs, the least indels, those are probably most closely related. So in this case, we have an indel, and it's most likely this is actually deletion, right? Because 2 of the sequences have the A and 1 does not. We have a SNP, and in this case, the mutation is probably the T to G, because 2 have the T and only 1 has the G. And then here we have another indel, but in this time it's probably an insertion in the second sequence, because both of these do not have, or this is the one that has an extra nucleotide in there. So when you're looking at a phylogenetic tree that has been constructed, whether it's by molecular or morphological data, you can evaluate whether the way we group organisms is valid or not evolutionarily by seeing if they are in a clade. So a clade is any group that contains the common ancestor and all the descendants. And basically what you're looking at is that you want to cut once to get that entire group off of the tree. So if I told you this green is a group we call animals, let's say, and I want to know, is animals a clade? Then you say, well, to get all the animals off the tree, we only have to cut once, which means that yes, it would be considered a clade. So when a clade is also called a monophyletic group, which are the most valid trees evolutionarily. Here's one SNP, here's another one. So here's the common ancestor and all its descendants in this blue box. Single SNP to get the blue box off of there, so that's a clade. Those are both monophyletic. Then there are 2 scenarios that are not monophyletic. So you see here, in order to get the red box, so all 3 of these organisms off, we have to cut twice. So that is not monophyletic. And in this case, let's say the orange box we call something. In order to get that off, you cut here, but then you also have to cut again because you're not counting whatever that organism on that last branch is. So there's 2 names for these types of groupings. One is called a polyphyletic group. So polyphyletic means that it's not including the common ancestor, or the RCA, the recent common ancestor. So of these 3 groups here, if we trace it back, the most recent common ancestor they share is right there, and that's not included, so that would be polyphyletic. A paraphyletic group is one where you're leaving one or more of the descendants out. So Here's the common ancestor, and in order to be a clade, it has to include all the descendants, but in this case, you've left this one out. And what we want is we want our namings of groups to match up with monophyletic groups on a tree. A clade is a very general term, really, because they nest inside each other. So it's not like phylum where you know what level it's at. It just, you can see here, the yellow is a clade, and that is inside a bigger clade, and that's inside an even bigger clade. In fact, this entire tree is a clade. And here's a real example of vertebrates. So you can see the different boxes. And the biggest clade that we're showing here are the vertebrates. So all the organisms and the common ancestor are included. Then the next one they're labeling here is tetrapods. So if we look with tetrapods, it's going to be this common ancestor and all of its descendants. So that would mean perch and lamprey are not tetrapods, which is valid. So you cut just once here to get the tetrapods off the tree. And so we'll learn the names of a bunch of these different levels as we go through the semester. So there are two terms that we use when we're describing characters on a tree. One is called a shared ancestral character, or plasiomorphy. Our book uses shared ancestral character. And that means it's a characteristic that was around before the clade split off. So it's not something unique to that clade. There are other groups that have that characteristic as well. And then the other one that we're more concerned with generally is the shared derived character, or synapomorphy. And so this is a characteristic that's unique to a group that distinguishes it and arose in the common ancestor of that group. So depending on what you're talking about, a characteristic is not intrinsically shared, derived, or shared ancestral. It depends on what clade you're talking about. So let's take a look at an example here. All right, if we are looking at the mammals on this phylogeny, which are the mouse and the chimpanzee, one characteristic that they both share is hair. And hair is unique to the mammal group, the mammal clade. Hair is a shared derived character, or synapomorphy. It's the characteristic that makes that group special. Something else that both mice and chimpanzees have are 4 limbs. But so do all these other groups, the salamander, the lizard, the crocodile, and the pigeon. So that characteristic arose back here. So for the mouse and the chimpanzee, that 4-limb characteristic is a shared ancestral character, or plasiomorphy. I will give you some sort of character table like this, and sometimes I use ones with zeros and ones, minus and plus. You could figure that out. So a minus in this one means that it does not have the characteristic, and a plus means it does. And so here are these derived traits. So those would be the synapomorphies. And then we have the different taxa here. And you'll notice that there's one group, the lampreys, which don't have any of these shared derived characters, and that's on purpose, picking an organism like that. It's known that lampreys are closely related to the rest of these, but they don't have any of the characteristics we're looking at. So we call that an outgroup. So we know the outgroup must branch first. So however you end up drawing this tree, you know that the first branch off of it is going to be the lamprey, and then everything else will be off the other branch. Okay, and so what you do is you try to figure out a tree that has the least amount of changes, the maximum parsimony. And so for example, if you look here, Jaws is in all the different groups. So that means that Jaws arose very early. In fact, Jaws is probably the first characteristic that distinguishes the rest of the group from the lamprey. So if we were drawing that, we'd be drawing the rest of the group, and we put the synapomorphy Jaws in the phylogeny. The lamprey does not have it, it arose, but then everybody else afterwards has it. And so you do the same thing for all of these. Now you'll notice some of them, it's not always all the way across, it just depends. Like these 3 have keratinous scales, which probably means that they are more closely related to each other, so maybe they group together. So let's look at building this phylogeny. Here's the common ancestor, it's a rooted tree. There's your lamprey, and we already know it's the outgroup. So there's our outgroup. The earliest branch here, this is the common ancestor of lamprey and everything else. The in group is everything else. And the first group to branch off of that's the perch. And the derived traits, remember I said the jaws? Everybody has jaws except for the lamprey. Turns out if you look at the character chart, everybody else has lungs except for the perch. And you keep on doing this sort of branching pattern, putting the synapomorphies on the lines and your organisms at the tips. So salamander here, claws or nails. And then we have a branch. Now in this case, they gave us derived characters of both sides. Now, of course, it just depends on what characteristics and which organisms are given. So it's not like there's nothing on this lizard line that's a synapomorphy. There's lots of things that are unique for the lizard group. We just don't have them listed on our tree. And so in the end, this is what our tree looks like with the least amount of changes. And you can see the synapomorphies are put on the lines, and then the branching pattern is what you're looking at. So even if the chimpanzee was next to the salamander, which it could be if you rotated around this node and you just flipped that up and swung this down, it's the same branching. So it doesn't matter that the salamander and the lizard are next to each other. That has no indication of relatedness, because we could have drawn the lizard in the middle of here. We could have drawn the lizard at the bottom. It just depends on the branching. There's a number of reasons you use phylogenies, obviously evolutionary relationships, but they actually use them in public medicine a lot. And in this case, they actually use it in a combination of medicine and forensic investigations, a criminal trial. So there's a physician, and he was accused of trying to kill his ex-girlfriend by injecting her with HIV-positive blood. But he said, of course, something like, well, she could have gotten that from anywhere. So they went about showing that the probability of that blood coming from the blood that the physician had access to was very high. And the way they did that was they built this phylogeny. So all of these endpoints here are different viruses, different HIV samples. And what they noticed here is when they did the phylogenies, the ones that the girlfriend, the types of sequences in her virus, were most closely related, most similar to the sequences in the viruses from that doctor's patients. And they were less closely related to samples of the virus from others in the community. You might notice here that this looks a little different, where we didn't draw all the branches the same length. And that's just because the branch length is representing something in this phylogeny. So it could be the amount of divergence, meaning how much sequence change there is. So a short line means not a lot of sequence change. A long line means a whole lot of sequence change. That's my guess here. But when we draw phylogenies, typically we line up everything on the end, because that just means present day and all we're looking at is branching pattern. Some phylogenies can actually show the amount of time that it has been since a divergence in the past. In other words, they can estimate the branch points on a tree. One of the ways we estimate the amount of time since divergence is by using molecular clocks. These are genes that we've chosen because they evolve at relatively constant rates, meaning that they accumulate mutations, SNPs, let's say, or maybe indels, at a given frequency. And so that allows us to estimate how long two groups have been diverging. There are different genes you want to look at for different lengths of time. Some genes, like the rRNA genes, so those are genes in the DNA genome, in the nucleus of the cell, that code for the little ribosomal RNAs that will go in the ribosome. Those genes evolve very slowly, because if you have mutations to the rRNA, it can't catalyze the protein building anymore. That's its job in the ribosome, helps put those amino acids together, and the organism dies. So it's very infrequent that a mutation occurs that is not going to kill the organism. So it's a very slow evolving gene. So when we want to look at events that happened very long time ago, and we're talking things like the divergence of bacteria and eukarya or something like that, we use rRNA genes. On the other hand, if we want to look at recent divergences, we need to look at genes that evolve more quickly. Some of those genes that are commonly chosen are mitochondrial RNA genes. So that is DNA in the mitochondria. So this is that organelle. So there's the DNA that it contains, and it has genes on it. Some of those code for the mitochondrial RNA. So those evolved more quickly. Selection pressure to stay the same is not as high. You have to figure out how fast a gene seems to be evolving. And basically what you do is you look at the number of changes and match that up with the fossil record or other groups that you already know. And then it gives you a slope of a line that tells you basically how fast the gene is on average changing. You don't want to choose a molecular clock that evolves very quickly due to natural selection, because that means some groups are going to be accumulating mutations very quickly and some groups are not. And so it could give you a false, you could think that those organisms diverged much longer ago than they actually did because one group has changed a lot because it's been under strong selective pressure. So sometimes that happens. So you're really looking for neutral mutations that don't confer an advantage or disadvantage for the organism. Sometimes this is not the case. Like I mentioned, natural selection can make clocks not run smoothly. So one of the things they use is sometimes multiple genes to try to help the estimates of a length of time. And all this information that we're getting with the DNA and molecular clocks and all that kind of thing is helping us to build a tree of life. And I just wanted to show you a little bit of what I'm calling the evolution of the tree of life and how it's sort of progressed through the years. So when we're back in the mid-1700s, Carolus Linnaeus, the same guy that did that hierarchy, kingdom, phylum, class, order thing, he split everything into two kingdoms. It was either an animal or it was a plant. And you'd be amazed about how many people actually think that this is how organisms are classified. Basically, it was like, do you move? Then you're an animal. Do you not move? Then you're a plant. Here's actually a page from his one of his publications. So this is animals, and you can see he's subdivided the animals into other groups. There's amphibia, Pisces, I imagine that means fish, insects, and then within that he subdivided again, and so this is showing his understanding of the tree of life. Now he, though, remember, he thought it was more like a ladder, not really a tree. Then we get to Ernst Haeckel. So he was in the mid-1800s, and he took into account that there were microscopic organisms, and he put all those microscopic organisms in protists, and then called this group Monera the ancestor of everything else. So Monera were these really tiny single-celled organisms that weren't very well understood. He thought, well, these are super simple. They must be the ancestor that gave rise to everything. So he basically had three kingdoms. You've got the animals, the plants, and then all the little stuff, which are called protists. Here's his actual publication. You can see here, Plantae, Protista, and Animalia. And there's the Moneres, way down there. And then you can see his divergencies. This is one of those trees that's a little confusing based off of how he's drawn it, but you can kind of get the idea. At least he's thinking like a tree now. Then in about in 1969, Robert Whittaker came up with the 5-kingdom grouping, and that's something that a lot of people are really familiar with still today, which we actually don't use that quite permutation anymore. But his groups were animals, plants, and fungi. So basically he noticed that fungi didn't photosynthesize like plants. So he's like, these should be in a different category because they don't get their energy the same way. Then he did protists here, which are the little organisms. They knew that there were cells that were with nuclei and cells without nuclei, so prokaryotic, eukaryotic cells. So he took that into account. So anything that was prokaryotic was Monera, and then anything that was eukaryotic and small basically was Protista. So that's the 5-kingdom. And here's his publication, and you can see there. And like I mentioned, he's looking at how they feed as one of his major criteria. And then in the 90s, and this work started in the 70s actually, but the publication we're going to look at was by Carl Woese, And he actually looked at gene sequences from many organisms, and including things just in the soil. And he looked at our RNA genes to look for really ancient divergencies. And what he noticed was there was this other group here that used to be grouped with the prokaryotes and Monera, but it was really different genetically. And actually the cells were somewhat different as well. So it's prokaryotic, but it's not the same as the prokaryotic bacteria. And so what he did is branched it into what we're going to call 3 domains. So you need to know those. There's the bacteria domain, there's the archaea, that's how you say it, archaea domain, and then there's the eukarya domain. And then within the eukaryotes, or eukarya domain, then you have the fungi, animals, plants, and then protists, which is not a great group, and I'll show you why in a second. And here's his publication from 1990, where he's actually showing this new group, archaea. And if you'll notice this branching pattern, we have bacteria, we have eukarya, and archaea. This is the same thing, right? So we walk along, bacteria branches off first, walk along, bacteria branches off first, and then we keep walking, and then archaea and eukarya branch. And so what he's actually suggesting here is that archaea and eukarya are more closely related than bacteria. And so that's the kind of tree that we look at today. And here's another version of that tree. You see here, right? So archaea and eukarya and bacteria. So let's trace it back. So domain archaea, here's the last common ancestor with eukarya. And here's the last common ancestor with bacteria. And we can call that the common ancestor of all life. And you can see this is not showing everything that exists, but many groups. Here are like the classic kingdoms of eukarya, right? Plants, fungi, and animals. They are primarily, if not completely, multicellular. And then we have some multicellularity in some of the algae. But besides that, everything else is unicellular, interestingly enough. And what you'll notice of why protist is not a good group, because if we were trying to use the word protist, let's look what we'd have to look at. Okay, these would all be considered protists. This would be a protist. And actually, there's other groups that are in there too. So look at this. We're missing, it's not monophyletic because we have green algae, but we don't have land plants. So both of those would have to be in the group to call it monophyletic because they have a common ancestor here. We have amoeba, but not fungi or animals, and they all share a common ancestor. So this is not a good group. It's a polyphyletic group when you use the word protista. So we just call them generally protists, and it's always lowercase because it's not an official technical name. And it actually probably gets more complicated. So here is from 2016, a published Tree of Life, one of the newest ones, and I'll show you here. Let's see. So we've got domain bacteria. So that's actually all of this is all what we're considering bacteria. And they don't actually think that they had sampling preference. They sampled soil and hydrothermal vents and in the salt, the saltwater Great Salt Lake or something like that, and in all sorts of areas, as well as just published sequences of dogs and different types of mushrooms and euglena, which is a protist. So they sampled a lot. So in fact, all of these, you'll see the red dots. That means that there are no cultured organisms that we have of those types. So they know that they're there because they got their DNA sequences from the environment, but they have not ever grown them and seen them as an individual organism. So this is an entire clade here that's unexplored. So that was pretty interesting. Then we have the archaea here, and then here is just the eukaryotes, which we knew was small. We wouldn't expect it to be much bigger. This is an unrooted tree, so it doesn't actually show the last common ancestor, but it does show the branching within those groups. Here's the same tree, but they've drawn it with a root, and this is where we see that the eukarya, different color, eukarya here, nest within the archaea. So they're more closely related to archaea than they are to bacteria, and there's that unexplored group that's monophyletic, actually. If you go back, and this whole group traces back to this last common ancestor. So that monophyletic clade is something that's quite interesting. And really, it probably gets a lot more complicated because there were events in the past, and still some happening in the present, of where the DNA of two organisms actually mixes. And I'm not talking about in sexual reproduction. I'm talking about like two single cells exchange DNA, or one cell picks up DNA from the environment and incorporates it into its genome, or two cells fuse together to make a whole new organism. So there's a number of ways that this occurs, but one of the classic examples here is this line, which is what we call an endosymbiotic event. You should kind of remember that from Bio 1 maybe. But the point here is that a bacteria was actually engulfed by a eukaryotic cell and then became part of all the eukaryotes after that, all their cells. So that was it's actually a bacterium that turned into the mitochondria. And then you'll notice here, same thing, there was a cyanobacterium that got engulfed by one lineage or a few lineages of the eukarya, and that actually turned into chloroplast in plants and some types of protists. And so these kinds of events, and then there's all sorts of exchange happening here in ancient times, so it makes it complicated to actually make a clean branching tree when you go really far back. So the tree of life is always evolving, and we're thinking, should we draw it as a tangled web? Should we even have a root on it? But new information is always helping us build more accurate trees, and we will use these throughout the entire semester.